Well, good morning, everyone, and welcome to the fall group meeting, and in particular, the SWOG Barlogie-Salmon Myeloma Committee. Hope everybody is having a happy and healthy and safe fall or late summer, depending on what part of the country you're in. And it's a great privilege to be able to present to you all of the great work that the entire team is doing. Just as an update for you, this is an overview of the leadership, and most, if not all, of the people are on the call. I'm privileged to be the chair. We have as vice chairs Brian Dury and Saad Usmani. Susan O'Brien is our executive officer. Our main stats people are Antje Herring, Rachel Sexton, and Adam Rosenthal. And moving onward, from a translational medicine perspective, Brian Walker, radiation, it's Chelsea Pinnix. We have a surgery contact. These are our imaging people. Pathology is still to be determined, and early therapeutics. And we also have, of course, digital engagement, an NCORP representative, data coordinators, and a big shout out, as always, to Jack Aiello, who's our patient advocate. This is an overview of our current enrollment, and you can see a very nice upswing over the last couple of years, which I think as you go into 2022, we'll probably find that those numbers continue to increase based on some of the currently ongoing and hopefully soon to be open studies. But I did want to thank everybody who has been enrolling, and also let you know that many of these studies still have a good ways to go, so that you would have an opportunity to open them and hopefully make good contributions. As you would expect, most of the enrollment is for our large maintenance study looking at lenalidomide versus lenalidomide plus daratumumab, and you'll be able to see that in more detail a little bit later. I did also want to acknowledge that the SWOG folks are making a contribution to some of the other cooperative group studies, including especially the Alliance and ECOG studies mentioned here. And you'll hear a little bit more about other opportunities that you have in this regard. And please do not forget that you can enroll to those studies as well. So as always, the agenda will look at recently closed studies, then those that are currently enrolling. We'll have a follow-up on some proposed trials, and then we'll show you some of the studies that are currently in the concept stage, but that hopefully will be available soon for you to open. So starting with the recently closed studies, and my apologies, since we do have a large agenda, I'm going to go through these fairly quickly because you've probably heard about the results of these studies before. But the previous large induction trial led by Brian Dury was SO777, which of course looked at lenalidomide and low-dose dex as the standard versus bortezomib len and low-dose dex as the experimental arm. Once again, nice enrollment from many sites. And you probably know the most recent update that was published last year looking at long-term follow-up from this trial. And just to show you quickly, these are the response data, which of course showed better response overall and better response quality for the triplet arm versus the doublet arm. And these are the progression-free survival with the triplet in red. And these are the PFS landmark analysis at 6 months, which again shows that there is an improvement. And then duration of response as shown here. Once again, VRD beats out RD, as you probably have expected and would know, and overall survival is better as well. Do remember that patients at most were getting 8 cycles of bortezomib, and then the two arms essentially both had len and dex. So it is a testament to the fact that better induction up front results in better outcomes out back. And this is overall survival by age, showing that the triplet was of benefit in both younger as well as more mature patients with myeloma. And this is at 12 months, which shows you that quality of response matters, with the VGPR group in green doing the best. There were some second cancers that were reported, although the numbers were not that dissimilar between RD and VRD. So at least encouraging that the addition of bortezomib didn't seem to have an impact. Next, in terms of recently closed studies, was the S1304 trial looking at 2 doses of carfilzomib, and both with dexamethasone, and this was led by Sikander Ailawadi. You may remember that this was at that time a 12-month regimen of carfilzomib, because that was what was FDA approved, and it looked at 20 per meter squared followed by 27 versus 20 or 56. Again, nice registration from all around the group, and this did lead to a publication last year in Clinical Cancer Research, which is now a very good over 10 impact factor journal. And just getting briefly to the response data, you can see here that although there was maybe a little bit of a trend towards a better response quality in arm 2 with a higher VGPR rate, the progression-free survival was not different in the 2 arms. And there also was no significant difference based on the number of prior lines, because we had allowed 1 to 6 prior lines of therapy here. In terms of prior bortezomib, you can see the data here. There was a suggestion that maybe with the high dose arm, there was a shorter benefit in patients who had bortezomib refractory disease, but probably this analysis was underpowered to make that conclusion. And here are the overall survival data, which again did not show a significant difference, nor by number of prior lines of therapy or prior bortezomib. Next, we'll get to S1211, and this was Saad Usmani's trial looking at high-risk disease. And of course, you remember this was RVD induction followed by RVD maintenance versus RVD ELO induction and then RVD ELO maintenance. Again, accrual from throughout the SWOG network as well as the NCTN partners. And Saad was able to get this published very nicely, took a lot of work on his part. And you probably remember that getting to the progression-free survival data, this was a study where we had hoped to show a large difference and a large hazard ratio with an improvement in PFS. And unfortunately, elotuzumab did not improve PFS in patients with high-risk disease, and there was no significant difference in overall survival either. Although I think, Saad, correct me if I'm wrong, but you've got a follow-up with more mature overall survival data where you're preparing the manuscript. Is that right?
That's correct, Bob. So, we're hoping that we can get that final publication out sometime in the first half of next year, and potentially an abstract at a national congress.
Excellent. Sounds very good. Other studies that we participated in included the study for smoldering myeloma, looking at lenalidomide versus observation, and Madhav Dodapkar, who at that time was part of SWOG, was our champion for this. And you probably remember this was observation versus lenalidomide alone, no dexamethasone. Again, decent accrual from SWOG, and this also was published. And in JCO, you probably remember the data showing that len as a single agent had a reasonable response rate, but most dramatically had a prolonged progression-free survival and reduced the risk of progression to symptomatic disease compared with observation. Here is the other way around, if you will, the cumulative incidence of progression, which was much higher if you just had observation in red versus len in blue. It seemed to be true across many subgroups. And this is in high-risk patients where there also was a difference favoring lenalidomide over observation. And the results were a little less obvious in intermediate risk. It'll be interesting to see what they look like with longer follow-up to see if perhaps there may be a difference that eventually emerges, although do note at the bottom that the number of intermediate risk patients was relatively small. And there were a few low-risk patients that snuck in, and they also seem to have a trend favoring lenalidomide, although it'll take, again, a longer period before we see this reach statistical significance. And the overall survival data have so far not been different, although note that they only go out to 3 years here. And of course, given how long patients with myeloma now live, it may take quite a while before we see a difference, assuming that we would be seeing a difference eventually. We also had a good participation in E1A11, which was the ECOG endurance trial looking at VRD versus KRD, and Jeffrey Zonder was our champion here. You probably remember this was for transplant ineligible patients or those willing to delay transplant, and a randomization upfront to VRD versus KRD, and then a second randomization either to 2 years of len maintenance or to len maintenance until progression. So once again, we had a very nice contribution here. Thank you to all of you that enrolled patients on this study. And this also was published very recently in Lancet Oncology. Once again, you probably remember, if we move forward to the data, actually, let me go back here to the response data. You can see, perhaps somewhat surprisingly, that the KRD arm with carfilzomib did not have a substantially different overall and complete response rate. I would have thought that it would have, but it did not. And these are the PFS data and the OS data. Again, the OS data are still early and we may see a difference eventually, but so far it looks unlikely. Also recently closing is the S1702 study, which was isatuximab for patients with previously treated AL amyloid. And Terry Parker from Yale was the study principal investigator. I don't know if Terry is on the line. If you are, would you like to take over?
Sure, thank you so much. So as stated, this is S1702, which was a phase 2 study of isatuximab for patients with previously treated AL amyloid. So it was activated in March of 2018. Our first patient accrued in June of 2018, and we had a great accrual, closing to accrual in September 2019. The initial accrual goal was 35, and the actual ended up being 43. And just to remind everyone of the study design, it was for patients with relapsed or refractory AL amyloid who had at least one or more prior lines of therapy and at least one organ system involved. Key inclusion/exclusion is that patients had to not be refractory to daratumumab. Prior exposure was allowed as long as it was 56 days previously. Performance status 0 to 2, adequate organ function as seen here. All patients received isatuximab, cycle 1 dosed weekly at 20 mg per kg IV, cycles 2 through 24, days 1 and 15. Primary endpoint was hematological response with several secondary endpoints, including organ response, safety, progression-free survival, and overall survival. So just kind of no real new updates, but we did present results of patient characteristics and the preliminary safety or toxicity data. It was an oral abstract at the International Symposium on Amyloid that was September 18th of last year. And then the primary objective of hematological response in addition to secondary objectives, was presented as an oral abstract at the American Society of Hematology annual meeting last December of 2020. What we did was present was that the overall hematological response rate was 77%. So kind of more recent updates is the remaining patients should have all completed the planned 24 cycles as of this October. We are waiting for the sites to complete the off-treatment forms and to update that final data. So our next steps will be to, again, update the previously reported hematological response rate, toxicity, and secondary endpoints, review the organ data, which we have not yet looked at, and then manuscript preparation with a publication goal of early first half of 2022. So that's all I have, so thank you.
So I think we're all looking forward to seeing the hematologic and organ response rates combined. And I think that'll be an exciting patient population and an exciting data set.
Yes, definitely.
All right. Well, thanks very much, Terry, for your leadership on this. And I think this was the first amyloid study through the cooperative group network in many years. And I think it did show that we can enroll well into this patient population. So I think that sets the table for some other amyloid studies, which you'll hear about later in this session. At this point, we are going to move to currently enrolling studies, which are predominantly really our maintenance study. And Amrita Krishnan at City of Hope is the lead here. Amrita, do you want to take over if you're on the line? Okay, that may be that she's not available. I'll take over unless one of you others, I don't think we have Hari on the line either. Oh, okay. Patrick sent the message that Amrita is on vacation, so hopefully she's enjoying and I'm sure well-deserved time off. So here's the little background and the current status of maintenance therapy after autologous transplant. Most of you probably do lenalidomide as a single agent, and that's based on the well-known CALGB and IFM studies, and it does have FDA approval. And of course, you know that daratumumab is used in pretty much every setting in myeloma, and soon to be in the smoldering setting probably as well. And there's very nice synergy between the two. So that essentially forms the background for the proposal to try to do len in combination with daratumumab to see if we might be able to push out the progression-free survival to a longer level. And we were able to get subcutaneous daratumumab provided for this study. This in part also builds on the experience from the Cassiopeia trial that was done predominantly in Europe. And of course, they looked at bortezomib Thaldex or VTD versus Dara VTD. Then patients got transplanted. They got a couple more cycles of consolidation of the same agents they had for induction, and then went to either Dara or to observation. And you probably know that the group that got the Dara maintenance had a better outcome. Also, there was the Perseus study, which is ongoing, which is Dara VRD versus VRD. And there is the Auriga study, which is for patients that are MRD positive after stem cell transplant. So this is the design of our study. You can see it's a randomization where you get either lenalidomide alone or lenalidomide followed by daratumumab. And the intent is for 2 years of maintenance and then to have an MRD assessment. If patients are MRD positive, then they continue on the same maintenance to which they've been assigned. If they are MRD negative, then there is a randomization either to stop whatever maintenance they were assigned to or to continue with that maintenance. The goal is to have 950 eligible patients, and so 1,100 is going to be the number we're looking to register and screen. The primary endpoint here is overall survival, and so there is 84% power to detect an increase in median overall survival from 10 to 15.7 years. And some of you who have been in the myeloma field for a longer period probably remember that once upon a time, the median overall survival for all myeloma patients used to be about 2 to 3 years. So that really tells you how much of an impact we've already had. And hopefully this trial will continue to add to that. There are interim analyses, as you can see, when there have been 25, 50, and 75% of the events. And the primary overall survival analysis will be at about 10 years. And there are key secondary endpoints, including PFS, clinical response, and MRD. This is the accrual update as of September, with 608 patients registered and 533 patients randomized. So this is really great news because we're about halfway there. And this is with substantially less than half of the timeline through. So we're far ahead of schedule in terms of enrollment, which I think is a testament to everybody's excitement and to the hard work of the team. This is just an eligibility update, and in particular, some information about COVID. And this was predominantly shared at the previous meeting. If you have any questions about that, please email either Amrita or people like Saad or I, and we can send you this information. This is just an overview of the study team. And I did want to give you a more detailed look at some of the information that Antje was able to pull on this. This is accrual by sites, so we can see nice contributions from a number of centers, quite a few places that have 10 or more, and hopefully more of you will be able to get into that range. This is just an overview of the patients that have been randomized so far. You can see median age in the low 60s, which is probably pretty standard for people that have undergone a transplant. A nice representation of males and females. We do have some Hispanics, and then we have a good representation of African Americans, which looks like it roughly parallels the proportion of African Americans in the population as a whole. And so that will be a nice result as it hopefully continues. And we do have a few other races as well. Here you can see the RISS stage distribution, with about 16, 17% of patients being stage 3, which also is not too far off what we typically see. So there may be some ability to determine whether high risk, as defined by RISS stage 3, benefit from LEN and DARA. As we would expect, most of these patients had proteasome inhibitor-based therapy at baseline, and almost all of them had VGPR or better at the time of transplant. Treatment summary is shown here. So we've had a few patients who have come off of the study, but a relatively low number of protocol deviations considering the large number of patients and the long duration of therapy. And fortunately, so far, only 1 death and only 11 progression events. Hopefully both of those numbers will stay low as we move forward. And this is just a quick overview of adverse events, with LEN in the middle column and LEN and DARA on the right. At least according to my eye, I didn't see any big differences except where you would expect. which include a slightly higher risk of infections with daratumumab, and that's certainly a known factor with any of the anti-CD38s. And here you can see also other issues, which again seem to be fairly well balanced. I've also put into the currently enrolling category the study for untreated Waldenstrom, because we're hopeful that this will be available very, very, very soon. And Sikander, you're probably on the line, so I'll turn it over to you to talk about the Vibrant study. Oh, okay. And Patrick tells me that Sikander is in clinic. Patrick seems to know everything today, so appreciate that. So this is a study for previously untreated Waldenstrom, as you can see from the title. And the objective is to determine the complete response rate in the two arms, with the standard of care being ibrutinib and rituximab versus the experimental arm, which is venetoclax with ibrutinib and rituximab. And you probably know that CR rates with doublets in Waldenstrom are relatively low, and hopefully we'll be able to see an improvement with that. Secondary endpoints include overall response rate, progression-free survival, safety, median time to best response, overall response rate, and then there are a number of correlatives that are included as well. And Dana Sparks, please note, in the chat function, has sent the message to everyone that hopefully this will be reactivated by tomorrow. So that'll be really exciting. You probably know that ibrutinib is FDA approved for Waldenstrom and is commonly used in the community because it's an oral and convenient drug. Rituximab is often added to it based on the Innovate trial that showed a significant advantage for the combination versus rituximab alone. And hopefully by adding a BCL2 inhibitor, because Waldenstrom has many aspects of its biology that are similar to that in CLL, we will see a significant increase in complete remissions and also in MRD negativity. And one feature to this study, which I think is also interesting, is that treatment is continued for 2 years and then stopped. And the hope is that we would get such deep responses that you would not need to continue therapy indefinitely, which at least so far has been the pattern for single agent ibrutinib. These are the eligibility criteria, which include newly diagnosed patients without prior systemic therapy, although single agent rituximab will be allowed as long as it was at least 1 year prior. Good ECOG study, no severe neuropathy, and then pretty standard creatinine clearance, liver function tests, and hematologic parameters. This is the treatment schema, so it's a 1-to-1 randomization comparing ibrutinib rituximab to ibrutinib rituximab and venetoclax. Again, after 2 years, if patients have had stable disease or better, they go on just to monitoring. And then for people who are on the ibrutinib rituximab arm, if they do progress, they have the option to add venetoclax and hopefully recapture a response. CR rate for IR was assumed to be 5%, and the hope is that the CR rate for the triplet will be 30%. We certainly would be not displeased if it were higher than that. And so we have a power of 80% with a significance level of 0.1 with 56 eligible patients to be able to meet those goals. And we are searching for opportunities to get extramural funding for correlatives, in particular to look at things such as the mechanisms of resistance and potentially to do single cell transcriptomics. Now we'll move on to some of the open ECOG studies that we are supporting. And the first one is the Optimum trial. the EAA 171 trial, and Christine Yee at Michigan is the SWOG champion. And Christine, if you're on the line, I'd be happy to turn this over to you.
Thank you, Bob. Can you hear me?
Yes, we can.
Oh, great. Thanks. It's my pleasure to serve as the champion for this study from ECOG group. As a background, MRD negativity has been translated into more favorable clinical outcome in myeloma patients. However, MRD status has not been used as the action decision-making in myeloma management yet. So this study, as a prospective study, tried to address the question whether or not we can utilize MRD status for patient myeloma treatment management decision by escalating the treatment, the post-transplant maintenance, adding Ninlaro to Revlimid, which is the standard of care in post-transplant setting. In patients eligible for transplant, currently the standard of care is single autologous stem cell transplant, followed by post-transplant maintenance, which is lenalidomide, also known as Revlimid. Progression-free survival median number is about 4 years, but we all know that patient can still relapse with appropriate maintenance therapy. Tandem transplant has been fallen off the favor, and also consolidation, there's no data to prove the benefit. So for this particular trial, we try to see whether or not MRD positive in the post-transplant setting, whether or not additional treatment, meaning escalating the treatment with addition of ixazomib, can benefit the patients. This is the schema for the trial. It is called Optimum trial, and there are 2 steps involved. Step 0 is the pre-registration, when MRD will be tested. So patients on lenalidomide maintenance therapy between 10 to 15 months of the maintenance therapy will be considered for this study, and the transplant has to be within 12 months of the diagnosis. Once the MRD is tested, MRD positive patients will be randomized to arm A versus arm B. Arm B is the standard of care arm with the lenalidomide 10 milligrams daily, days 1 to 28. Placebo will be also utilized on days 1, 8, and 15 to match the study arm, which is arm A, with lenalidomide plus Ninlaro 4 milligrams days 1, 8, and 15. And both arms will be continued until disease progression or unacceptable toxicity. So for this particular study, up to 840 patients will be screened, and about 500 patients will be randomized at step 1. And the treatment will be blinded because of the placebo was set up in the standard of care arm B, and patients with creatinine clearance 30 to 60, those patients' lenalidomide dose will be reduced to 5 milligrams. So the primary objective for this particular study is to assess the overall survival in those patients who is MRD positive after 1 year of post-transplant maintenance with lenalidomide alone. And for the study arm, we will add ixazomib versus the placebo in the control arm. The secondary objectives include the conversion of the MRD from positive to negative, and also to assess the PFS improvement after patients receive the additional Ninlaro on top of the lenalidomide if they are MRD positive after 1 year of the post-transplant maintenance, and also to assess the quality of life in those patients. Additional secondary objectives include the kinetics of the medullary and extramedullary relapses, and also to compare the MRD assessments, as well as to look at cell-free DNA as the marker for MRD in myeloma patients. The treatment, as mentioned earlier, those are standard of care lenalidomide 10 milligrams days 1 to 28 every 28-day cycle, and also Ninlaro 4 milligrams weekly days 1, 8, and 15, versus arm B, the same dosage and a dosing schedule for len and with placebo. So we mentioned that this study will enroll 510 patients for randomization, 1-to-1 randomization to arm A versus arm B. And it's about 80% of the power at one-sided, 2.5% is significance to detect a difference at 4-year overall survival from 60.4% to 72.2%, and which will reflect approximately 35% of the reduction in hazard. And also, the study will expect 714 patients to have MRD measured, which will result in 536 patients with MRD positivity. If there's any questions, please feel free to reach out. Thank you.
Christine, I did have a quick question because the topic of cell-free DNA measurements came up at yesterday's meeting of the clinical trial portfolio group. And who is doing these measurements in the context of the study?
I believe the correlative studies are being sent to a Mayo Clinic.
Gotcha. Okay, fair enough. Well, thanks very much. Any questions for Christine about this for now? Okay, if not, take note of her email and contact her if you have questions. Do please think about opening this study and enrolling patients. It does not overlap with the previous maintenance trial that you saw from SWOG because it enrolls patients that are already on maintenance. So if you've gotten somebody who was already started on treatment and can't go on that study, then this one would be a possibility. Also, if Christina Gowan is on the call, I would ask her to mention the equate trial, which she serves as the study champion for. All right, we'll take that to mean that Christina wasn't able to join. But this is an overview of the ECOG equate trial, which is a randomized phase 3 study for newly diagnosed patients with multiple myeloma who have no intent for early stem cell transplant. And what you can see is that it involves an induction therapy of 9 cycles with daratumumab, lenalidomide, and dexamethasone. Then there is MRD testing, and based on MRD status, there is then a randomization so that patients can either continue on to more therapy with DRD or to have bortezomib added for another 9 cycles, and then both of the groups get dara plus lenalidomide maintenance as progression. So essentially it's asking the question of whether adding a 4th drug for patients that have not achieved MRD negativity will be of benefit in long-term outcomes. You can see that the goal is to enroll 1,450 patients, and that stem cells can be collected after 4 cycles of therapy in case you want to consider doing transplant at the time of progression. This is a little bit of an update on the study activation, which occurred October 27th, so almost a year ago. There have been 479 sites that have applied to be part of it, and 456 of those have been approved. And there is enrollment from ECOG Akron, as well as Alliance, NRG, and SWOG. But this is still a very early trial, and so hopefully you will have interest in opening this study if you haven't yet, and in enrolling patients also. That brings us to a close on the currently enrolling studies, and now we have a section where there is follow-up on proposed trials. And you heard earlier about the previous iteration of the high-risk study, and we have a new version that is being worked on, and this is being worked on by Brea Laipi and Saad Usmani. Brea, are you available to speak about this? Or if not, Saad, could I ask you to pinch hit?
Certainly, Bob. So, this concept eventually, when it does, move forward through, the SWOG Executive Committee and CETA will probably change the name. But, essentially, this is a follow-up to the SWOG 1211 trial. Initially, the proposal for the 1911 study was the comparison of KRD with or without daratumumab in a randomized phase 3 study for newly diagnosed high-risk patients. But given the advances that have been made in early phase therapeutics, especially with bispecifics, this new concept has been developed by the SWOG Myeloma Committee. The primary objective we'll come to the design, but the primary objective for the randomized phase 3 is to actually show overall survival benefit in the experimental arm compared to the standard of care arm. The standard of care arm being considered is VRD with daratumumab, similar to how it's been utilized in the Griffin study, and there are randomized phase 3 Cepheus and Perseus that are ongoing. And in the experimental arm, the BCMA-directed bispecific antibody teclistamab would be added. The secondary endpoints will be PFS, overall response rates, and safety, and importantly, MRD negativity post-induction and during maintenance is going to be compared, as well as quality of life. Bob, if you can kindly move to the next slide. So, this is a very simplistic schema. In our initial conversations with CTEP, we may actually go to a randomized phase 2/3 design with this study. The induction phase will include 8 cycles of induction with VRD daratumumab, followed by dose attenuated maintenance with RV and daratumumab. And in the experimental arm, teclistamab will be added. Stem cell collection is permitted after 4 cycles of induction treatment, but stem cells will be deferred to the first relapse and beyond. The definition of high risk will include revised ISS stage 3, as well as certain high risk features that still fall under the revised ISS stage 2. But we recognize that these patients do behave like high risk and have earlier relapses. Then extramedullary myeloma, as well as circulating plasma cells of 5% or more, will be included in the high risk definition. Next slide. I think this might be it, Bob. This is a quick peek as the concept becomes more granular. We'll provide more information probably in the spring meeting next year in Seattle, I guess.
Very good. Any questions or suggestions? Feel free to bring them up either now or by email or at one of our monthly calls. As Saad mentioned, this is still a work in progress. And so if you have a very good idea about how we could improve this, there is time to be able to make the adjustment and hopefully maximize the value of the study. Yeah, it looks like there was discussion in the chat room about the decks in arm 2, and Saad, thanks for clarifying that question. And then finally, we'll get to the studies that are currently in the concept stage. And one of these is a study by Patrick Hagan and also from Patrick Stiff. And I know Patrick Stiff is on because he's been updating me about where the folks are. And either Patrick A or Patrick B, would one of you want to take over on the presentation
Well,
of this?
I don't know. I don't know which Patrick I am, A or B, but we'll that's have to be determined. So thanks, Dr. Orlowski.
We'll make
So-
you A since you're
Good,
volunteering
good.
to
I'm
present.
further away from retirement, that's for sure.
Ooh, okay.
No, Dr. Stiff's been great and helpful with this, but this I wouldn't really call this my study. This has been a labor of love with a lot of support with some of those listed here, in addition to some that are not. So this study is really looking to ask, what is the optimal consolidation approach in newly diagnosed transplant eligible systemic light chain amyloidosis patients? Particularly with some recent updates in therapeutics. This is a question we're asking ourselves in clinic now more and more. So what we're trying to ask is, after 2 cycles of DARA-BCD induction, is the optimal consolidation to continue with 4 additional cycles or to take patients to high-dose chemotherapy and stem cell transplant? So we can go to the next slide, I think. So, oh wow, these may be the wrong slides. Just in way of background, there's really only been one randomized study looking at the role of high-dose chemotherapy and stem cell transplant in amyloid, and it's an old study. It's outdated. Patients got induction with melphalan and then were randomized to transplant or not, with very high transplant-related mortality, so 24%. So we just don't see that in current practice. In kind of current practice, our TRM is much lower in the 2 to 5% in experienced centers. And then furthermore, induction therapy is really becoming the standard of care pre-transplant. So really this has been shown to consistently impact both PFS and overall survival, as well as actually the dose of melphalan that patients receive at time of transplant. And then in terms, I think, importantly, of the clinical importance of this question is, practice patterns are really quite dramatically different in the US compared to other parts of the world. So, the data is a little bit tough to get in terms of granularity, but probably about 1/3 of transplant eligible patients get upfront transplant in the US, and this number is much lower. The Alchemy study, which is a huge registry study, looked at 1,000 patients plus, and only 3% of those got an upfront transplant, and some of the other data from Europe is probably at the highest in the 5 to 10% range. And then finally, what the recently completed Andromeda study showed us, which looked at DARA-BCD versus just CyBorD induction, is that we can enroll lots of patients on amyloid trials. And as we talked about a little bit earlier, we actually showed that in SWOG recently with our isatuximab trial in the relapse setting. We enrolled very quickly, rapidly, quicker than we had even planned. So there seems to be an appetite, I think, to put patients on amyloid trials more so than there used to be. Go to the next slide. So these are, I apologize, I think these may be some older slides, but they'll get the point across. So importantly, transplant numbers are going up. So I don't have the 2020 updated data, but despite better induction therapies, we're doing a few more transplants each year. So again, we think in terms of feasibility that we're in a good place potentially to do the study at this time. We can go to the next slide. So essentially what we're asking is that we're hypothesizing that the clinical outcomes of patients treated with high-dose melphalan and autologous transplant as their consolidative therapy will be superior to those who would go on to just complete consolidative therapy with dara-BCD, with their primary outcome of interesting modified progression-free survival. And for those of us not as familiar, this is somewhat of a novel endpoint, but something that was agreed upon by our group as well as input from ECOG and others. And this is defined as deterioration in PFS due to either death, clinical manifestation of organ progression, so cardiac or renal failure, or the development of hematological progression. So that will be our primary objective, will be to compare the modified PFS between a high-dose melphalan arm as opposed to the dara-BCD arm. Importantly, there will be a lot of secondary objectives we want to look at, including quality of life, which we feel will be actually very impactful if the study ends up being a negative study where there's no difference in the primary outcome. Quality of life and toxicity from these therapies are quite different, with short-term toxicities from transplant, more long-term toxicities in terms of neuropathy, et cetera, from the Velcade. We'll also look at survival, treatment-related mortality, so not just transplant. We want to look at deaths on both the transplant and a non-transplant approach, because as anybody who treats amyloid knows, these patients can be more challenging, certainly compared to myeloma patients. Organ progression, hematological progression, including depth of response and MRD negativity. Go to the next slide. So this is the study schema. So patients will be enrolled based on eligibility, which I'll talk a little bit about coming up here. All patients, regardless of where they're randomized, will get 2 cycles of dara-BCD induction. If they're still deemed transplant eligible, they're randomized in a 1-to-1 to complete 4 additional cycles of dara-BCD consolidation versus high-dose melphalan. And then all patients will go on to complete a total of 2 years of daratumumab or 18 months of maintenance. Importantly, randomization will occur at the time of enrollment. One of the questions that is critical on this study is, patients who achieve quick, deep responses, which can be quite quick with dara-BCD, are those patients still eligible? Are those patients still going to benefit from transplant? So in order, we feel, to reduce potential dropout, patients will be randomized at time of enrollment. Go to the next slide. So kind of some key inclusion criteria. So again, transplant eligibility be both at time of induction, initiation of induction, and after the 2 cycles. Ultimately, it will be left up to the enrolling center, but we are going to strongly advise that centers follow the Mayo criteria, which essentially includes certain cardiac markers, which I'm more than willing to talk more about with people offline if they have questions. Also importantly, patients have to have at least a partial response to induction. This is going to be really rare, but maybe in the 5% range, patients who get dara-BCD induction don't achieve a partial response. So these are not patients we would want continuing on to the same therapy that is essentially being ineffective. Some key exclusions, so they cannot have symptomatic myeloma, although we won't exclude patients purely based on the plasma cell burden in the bone marrow. And then, as I said, patients need to achieve a partial remission. We can go to the next slide. So just some considerations. So again, about 250 to 300 transplants are being done annually. As I talked about, the overall response rate quite high on the dara-BCD arm. We'll go back one more. There we go. Why did we choose 2 cycles of induction? That's because median time to VGPR and CR in the dara-BCD and enrollment, it was 17 and 60 days respectively. So it was felt that probably no additional than 2 cycles were needed. We're estimating that about 5 to 10% of patients won't be randomized due to lack of response to induction, and potentially an additional 5 to 10% due to toxicity. Again, this is a critical question in amyloid. Many patients look very transplant eligible when we meet them initially, but as we start them on therapy, with subclinical organ involvement, some of these patients run into a lot of trouble. So again, we're going to lose some patients due to dropout from toxicity, a few due to lack of response, and this will be accounted for statistically. Go to the next slide. So I apologize. Some of this is the numbers here are right. Some of the statistics have been updated, but in the end, essentially what we're going to need to do is randomize 119 patients on each arm. So that will actually be allocated to treatment based on some of the issues I just spoke about on the last slide in terms of dropout due to toxicity or lack of response. We're accounting for about a 20% that will be ineligible at randomization, which puts our numbers just under 300 for enrollment. And we're looking to enroll about 10 patients per month. So we would expect this study to be able to enroll over about 2 years, potentially a little bit more. And then also importantly, there's a lot of interest here from the BMT-CTN myeloma intergroup. So a survey was sent out, which shows a lot of interest in this study in terms of feasibility and centers coming on board. We can go to the next slide. Oh, that was it, yeah. So I saw this is a little bit of an older slide. There are some updates to the statistics that are important, but overall don't change the major design. But so, you know, this is a study I'm certainly excited about. I think for SWOG and intergroup and amongst a lot of us around the country, this has the potential, in my opinion, to change standard of care, and is a really important clinical question that we're asking in our transplant clinics about these patients who are now achieving these nice responses with induction. So I'll kind of stop there, and I'm open for any questions or comments.
So Patrick A., which of the arms do you think will be better?
A little bit of a hammer and a nail. So I suspect that the high dose melphalan arm will be better in terms of long-term outcomes. You know, we may be surprised. I think right now we have decades of follow-up for high dose melphalan and amyloid, and only, you know, a couple of years of follow-up for DARA-VCD induction. And, you know, the big question here is long-term, what's best for these patients. So that's my general bias, but the shorter answer is we don't know. And when patients achieve CRs in 2 months of induction, you know, this is a really, I think, relevant question that we're asking with our patients.
Very good. Maybe we can.
I have a question.
Yes.
Why not allow patients, you know, who have received some previous therapy to go on to get enrolled at the time of randomization? So this will sort of, you know, make it easy for you to enroll patients who have, let's say, you know, already received one cycle of Cybo-D or DARA-Cybo-D, or a couple of cycles before.
Yeah, this has come up in conversation, and we've kind of gone back and forth. As it currently stands, we're not going to allow any initial therapy, but I think that, you know, as this study moves up through CTEP and larger steering committees, I'm sure this will come up again. I don't think we're going to lose a lot of patients, because generally patients who are transplant eligible, I would say, are a little bit less in crisis mode, but it certainly is something that we could potentially lose some patients. So not to mention, you know, just getting these patients referred into our centers, you know, a lot of these patients do start therapy in the community and then are referred in for a second opinion. So I think it's a great question. As of now, this is the way the study is designed, but you know, if we're having trouble with enrollment, it could be something that we could amend in the future. But as of now, that we're not allowing any initial therapy.
One thing we may need to talk about, there's some discussion in the field, I think, about whether patients with T1114 may do particularly well with CD38 therapy. So of course, both arms here get DARA, but it may be worth thinking about stratifying based on 1114 plus or minus in this study.
It's a good point. And, you know, we have three stratification, the ability with our updated numbers, which again, I apologize, weren't there. We have the ability to stratify for 3 factors. That's what I've been told. Right now, we have it based on response. So basically CR versus less than CR, and based on the Mayo stage. But it's something I think we could potentially stratify for, but we'd have to obviously just make sure in terms of our numbers. I don't think we can go up in terms of our numbers. I think we've kind of got our total enrollment goals at a reasonable place. We'll see again what others say when this moves up to the steering committees, but I think it's something, you know, in terms of enrollment, we're going to not be able to increase too much.
Yeah, the stratification shouldn't, and Antje can correct me if I'm wrong, because I'm a faux statistician, but I don't think the stratification should change the sample size.
All right, Bob. Yep.
Okay. I guess the only final thing I'll say is that, you know, we are going to look at 2 patient-related outcome assessments, both at induction, pre-consolidation and post-consolidation. That's going to be very important in terms of if it's a quote unquote negative study, because we could see big impacts and difference in terms of quality of life on these therapies. And then the translational piece of this is still a work in progress. So I have some thoughts. We're working on this, but if anybody in SWOG or outside has strong opinions or interest, I'm more than willing to have discussions, because that's a little bit more nascent in its development.
Okay, very good. Well, let's move on. And with that amyloid background again, we have another study that has been discussed and would be led by Divya Bhutani from Columbia, which is with CAEL-101. Somebody has to come up with a snazzy name for that. Divya, are you on the call perhaps to be able to present?
Yes, Dr. Orlowski. Is it okay
Take
if
it.
I share my screen? I just have some updated slides.
Sure. I will stop sharing and you can go for it. I would have probably screwed up forwarding them anyway, as I have for some of the other presenters already.
Thank you.
So when you all do my evaluation, feel free to say that he was a decent chair, but he couldn't advance slides correctly to save his life.
Can you see my slides?
Yes, you're good to
Okay.
go.
So this is a study that we are planning, which is going to be a double-blind randomized phase 2 trial of KEL-101 versus placebo in patients with primarily renal AL amyloidosis. So just a little bit of background, patients with advanced AL amyloid continue to have poor prognosis, although there have been major improvements, especially with the Andromeda trials. We see much higher response rates with the addition of daratumumab. This is the data that Mayo Clinic has published previously, showing that patients who are transplant ineligible, continue to have very poor survival, even with the improvements in therapy. The blue curve is the most recent time period where the 2-year overall survival is still very poor at only 47%. Furthermore, patients who are able to get hematologic responses to newer therapies continue to have delayed organ responses. So clearly there is a lot of room to improve organ responses in these patients. So our question we are asking is, can monoclonal antibodies targeting amyloid fibrils directly improve these outcomes? This is sort of, some of the data, as you guys know, that there was, previously there have been 2 drugs that have tried to do that, not with a lot of success, unfortunately. This was one of the trials that was done with a drug called NEOD-001 that was primarily focused at that time in patients with cardiac AL amyloid, in which patients who had previously been treated with antiplasma cell therapy were randomized to NEOD-001 versus placebo, with the primary endpoint being the cardiac response. And the study, as you guys know, was negative at that time. But interestingly, there was a sort of a significant, almost a 20% difference in the renal response rate in those patients. At the same time, another sort of a small trial was opened primarily in patients with the renal amyloid in Boston at Tufts University, but the trial was actually shut down in the beginning. And that trial, we have some data that there was 2 out of 6 patients actually had a major renal responses in that trial, and 1 patient surprisingly had a drop in 24-hour proteinuria from 17 gram to 4.9 gram after 3 months of therapy. So possibly, there is some signal that these drugs may have activity in renal patients. So this is the drug, that was initially developed at University of Tennessee. It's primarily an antibody which binds to a loop flip, that happens during the fibrillogenosis, while so the drug does not really bind to circulating light chains, but it binds primarily to the deposited light chains at this spot, at the loop flip. Previously, there has been published data that the drug is very well able to bind to amyloid deposits. This is a patient with primarily liver as well as bone marrow amyloid, and the drug showed, very good binding to the areas where the amyloid is deposited. This is the phase 1 trial that we did at Columbia, and this was just recently accepted for publication in Blood. And we showed about a 67%. This was only given for 4 weekly doses. And we showed a 67% cardiac response and close to a 50% renal response in these patients who had been previously treated. There is an ongoing phase 2 trial, primarily at Cleveland Clinic, that was presented as an abstract in ASH last year. And they had, at that time, enrolled a total of 13 patients, but 7 patients primarily with renal amyloid. And all 7 patients actually had a response at that time. And one patient, especially this patient, had close to about 80% reduction in proteinuria after being enrolled. This is obviously patients are getting a plasma cell therapy in addition. So this trial does not tell us, which one is driving what, but clearly there seems to be, a major sort of possible improvement in proteinuria. The drug is, very, very safe, and there was very few, phase 3 or higher adverse events, very well tolerated. So based on these, sort of we are, as you guys know, the company is already doing 2 trials in cardiac patients, but they are very interested in a renal concept. So we have sort of proposed this study of patients who have previously been treated and have achieved a CR or a VGPR after their anti-plasma cell therapy, but have persistent proteinuria of more than 0.5 grams per day. The patients will be randomized to KEL-101 versus placebo, and the treatment duration will be 12 months. And I will come to our primary objectives will be to compare the renal response rate in patients treated with KEL-101 versus placebo. We are looking at some secondary objectives, including cardiac response, organ progression-free survival, overall survival, safety, as well as the patient-reported outcomes. This is going to be the eligibility. So we are, I'm sorry, we have actually changed the degree of proteinuria to about 1 gram per day. Previous therapy with the plasma cell therapy, and a patient should be in a hematologic CR or a VGPR. We are allowing up to 2 prior lines of therapy, as long as the patient is within about 4 years of starting the first line therapy. We didn't want the patients to be, too far out from the start of therapy, since as they go along, they may sort of develop changes in the kidney that are not reversible. We will allow patients to continue CD38 maintenance therapy as per the discretion of the treating physician. Patients must have, patients must have completed their last dose at least 16 weeks prior to starting cycle 1, day 1. And for transplant patients, we want at least a 24-week duration from completion of the transplant. We are sort of, still discussing these timelines a little bit, but we wanted to sort of make sure that the patients on the placebo arm, do get some time to get their responses, because we do not want, the placebo arm to continue to have a, too high of a response rate if we enroll too quickly. As I said before, the CD38 antibody maintenance therapy can continue as per the discretion. And other than that, adequate, kidney, hematologic, and hepatic parameters, GFR more than 30. We will exclude patients who have, advanced heart disease with a NYHA class 3 or 4, NT-proBNP more than 8,500, or if they have previously received any other anti-amyloid antibody. So this is a double-blind randomized phase 2 trial with 1-to-1 randomization. The KEL-101 will be given at a 1,000 milligram per meter square weekly for first cycle, and then every 2 weeks for a total of 12 months. Our primary endpoint will be the renal response rate that we discussed, with a more than 30% reduction in proteinuria. And these are the secondary endpoints that we have already discussed. We sort of, anticipate that the placebo arm will also continue to have a renal responses. And we sort of hypothesis is there will be about a 30% response rate in the placebo arm, and that we will see a response rate of close to 50% in our experimental arm. So based on that, we have done the calculations that we will need close to about 120 patients, so about 63 patients per arm. We will do stratification by prior history of transplant, as well as DFLC of less than 1. And we will also perform interim fertility analysis when 50% of the patients have been treated for 6 months. we had some prior discussion on Tuesday, and right now, we are working on possibly allowing a crossover for patients in the placebo arm who have evidence of organ progression. In terms of patients who are having hematologic progression while on the study, we will allow them to restart anti-plasma cell therapy while they can continue on the trial. That's it. Yeah. Any questions?
One quick question that I hadn't thought about before in your presentations. You picked a 1-year duration of treatment, which was longer than what you had done in your own study. So I'm just wondering, why pick 1 year other than that it's a nice round figure?
Yeah, I mean, we haven't, yeah, that's a good question. I don't have an easy answer. I mean, that the one that we have done, the phase 1 trial, the treatment was given only for 4 weeks, was to primarily to establish safety. This is what they had done previously, in the other studies, I think the NEOD 001 study, and currently the ongoing CALM studies for cardiac patients are also giving a treatment of around 1 year. So this was sort of based on that. I think we think that, the longer you give the treatment, it does take some time, for the amyloid deposits to come out. So if you keep the treatment too short, you may not get enough sort of efficacy to get, obviously the protein out, which is a relatively slow process. So that is the thinking behind it. I don't think we have a much better sort of, rationale to come up with a duration. Yeah.
But let's say, for the sake of argument, that you have many patients, which hopefully you will, who have a big decrease in their proteinuria, or maybe even a reduction down to normal levels that happens within 3-4 months. Would you think that they need to continue on treatment, or might it be reasonable to, sort of the typical oncology thing, do 2 cycles past best response, and then stop? I know that, the first thing that you want to do is, of course, is establish that this scheme works before you start pulling back on it. But I just thought about that question and thought we could talk about it, even though neither one of us knows the right answer here.
Yeah, I think it would be hard to, change in mid-study. But I think if that happens, I guess we can discuss it at that point. But I think if the drug is so well tolerated, I see probably, unlikely that we will do any harm by giving a longer treatment, other than the, the patient sort of, convenience as well as cost, obviously will be one factor. But I think the chances that we will do any harm by giving longer treatment is unlikely.
No, I agree. It looks like a fairly non-toxic agent, although there's always the consideration for the inconvenience to patients as well as the cost of treatment. So I think I'm fine with it staying at 1 year, but just be prepared that somebody else will probably ask you why not do it for a shorter period, especially, let's say, in a patient who has gotten to MRD negativity with respect to the plasma cell component. And if you've shown that you've completely resolved their organ dysfunction, one could make an argument that maybe they're not going to lay down more amyloid if they're in MRD negativity with regard to the plasma cells, and that maybe a less than 1 year duration of treatment would be okay.
Sure. Yeah, we can definitely keep that in mind. Yeah.
Okay. Any other thoughts about this study or discussion, input, interest level, questions? All right. Well, very nice. And hopefully we'll be able to move that forward. Let me figure out where my slides are, and we'll go back to those. Here we go. And let's do this. All right. So we'll go through those slides. Okay. So next on the agenda is the currently S2209 numbered study, which is a phase 3 trial for newly diagnosed myeloma patients that are frail or transplant ineligible. And I think Brea or Christine, maybe one of you can field this and describe the study design and where we are with getting this trial hopefully approved and up and running.
I'm not sure if Brea is on the call. If she's not, I'll be happy to talk about this concept on behalf of our study team.
Yep, go for it, Christine. Hopefully I'll be able to get the slides right for you.
All right, thank you. So we actually introduced this concept in the last 2 SWOG meetings, and just to refresh everybody's mind, this study, we're trying to address the patient population who are frail and elderly patients who are not eligible for autologous stem cell transplant. We initially we were comparing the 2 most commonly used myeloma regimens, including RVD Light, which is based on a phase 2 study from MGH group, and also the other regimen, which is DARA-RD, based on the Maya clinical trial from Janssen. And the purpose of this study is to compare these 2 regimens. So we set up 2 primary study objectives. The first one is to compare the PFS in transplant ineligible newly diagnosed myeloma patients receive RVD Light induction followed by lenalidomide as maintenance therapy versus DARA-RD as induction followed by Revlimid as the maintenance therapy. And the second primary objective is to compare the overall survival between the arm of patients on, I'm sorry, the slide actually needs to be just modified a little bit, is to compare with arm 1, which is RVD Light followed by Revlimid maintenance versus DARA-RD as induction followed by the DARA plus Revlimid as the maintenance therapy. We have several secondary study objectives described here. One is to compare the PFS in the arm 1, which is the RVD Light followed by Revlimid maintenance versus versus DARA-RD induction followed by the DARA and Revlimid maintenance, which is arm 3. Sorry, I should have put the schema up front so that everybody can have a better idea. Arm 1, 2, and 3. Thank you, Bob. So yeah, because I think if we review the schema, probably it's easier because the study concept actually initially is just to compare arm 1 and arm 3. However, we got the feedback from NCI steering committee trying to clarify the question whether or not we are comparing induction versus maintenance therapy, because RVD Light followed by Rev, this commonly used regimen is different from the Maya regimen that is DRD as induction followed by the DR as maintenance. So based on this feedback, we actually modified and updated this study design. Now we have 3 arms. So arm 1 and 3 are the same as we initially proposed, but we added arm 2, which is to start with induction therapy, DRD, as induction regimen followed by Revlimid alone as the maintenance therapy. So now our comparison would be to compare arm 1 and arm 2 as for the PFS in the primary endpoint. And the second primary endpoint is to compare arm 1 versus arm 3 as the entire regimen, induction followed by maintenance therapy. So if Bob, you could please go back to our objectives. Great, thank you. For the major secondary study objectives, we will compare PFS in arm 1 versus 3, and also to compare the OS arm 1 versus arm 2. And we will also compare the ORR, overall response rate, in arm 1 against 2 and 3 respectively, and also to compare the safety of arm 1 versus arm 2 and 3. We will also describe the median time to response according to IMWG response criteria, CR or better, VGPR or better, or PR or better on all 3 study arms. Next slide, please. So the background I already mentioned, because of these 2 regimens are the most commonly used regimens, so we're trying to compare these 2 regimens which will provide better survival for our newly diagnosed multiple myeloma patients who are frail or elderly. Next slide, please. So the hypothesis is based on whether or not this DRD induction followed by the DR maintenance will offer longer progression-free survival in this patient population compared with RVD light followed by the Revlimid maintenance. And also we try to ask the other question is whether or not DRD followed by DR maintenance will offer longer overall survival in the same patient population compared with the RVD light followed by the Revlimid maintenance therapy. Next slide, please. Next slide, please. Yes. So for the key eligibility, certainly this reflects our study patient population. This is a frail patient, and we try to mimic the setting of the real world patient population. So as we know, the onset of the multiple myeloma, the age group is around the 70 to 75 years old. And many of them, they may already have comorbidities, pre-existing comorbidities. So we try to relax the inclusion criteria to make it similar to the real world patient population that we encounter in clinic on a daily basis. And we would encourage our community oncologists to enroll patients in their clinic. So this inclusion criteria reflects that concept, and then as we can see, we will use myeloma frailty index to include the patients either frail or intermediate frail based on those criteria. And also for organ functions and also marrow functions, we try to relax those parameters as well. So for example, for the kidney dysfunction, patients, if they have intermediate frail score, patients with creatinine clearance less than 30 will be included in the as the eligibility. Next slide, please. For the exclusion, that's pretty standard. This is only for frail and elderly multiple myeloma patients, so not including amyloidosis. We already have 2 wonderful concepts just presented. And also Waldenstrom, so we have a vibrant study and history of malignancy. Those are pretty standard for all the myeloma clinical trials. Next slide, please. In terms of the statistics, so our primary endpoints to match our primary objectives, we have a PFS to compare arm 1 versus arm 3, and also we have overall survival to I'm sorry, the PFS to compare arm 1 versus arm 2, and overall survival to compare arm 1 versus arm 3. And again, secondary endpoints are to match our secondary objectives. Next slide, please. We will also consider the risk stratification based on the myeloma frailty index score, and also revised ISS staging. So staging 1 and 2 versus 3. We based on the sample size calculation, and a huge thanks to Andre and Adams, our wonderful statisticians, to help us with this study concept and a stats design. So a total of 507 eligible patients to accrue about 170 patients for each arm, and then to account for a 10% eligibility rate, a total of 188 patients will be accrued per arm for a total of 564 patients. And the randomization will be 1:1:1 fashion among all those 3 study arms. We will have interim analysis built in as well at 3 different time points. In a first interim analysis, we'll test for futility only, while the second and third will test for futility as well as efficacy. Next slide, please. And we will welcome any input and comments or questions so that we can improve this study design. A good news to share is we actually have this study just approved by our SWOG executive committee, and now we have the new study ID, which is 2209. And we look forward to work with many other team members to move this study forward. Thank you.
Very good. Thank you, Christine. So hopefully this will soon be moving up to the myeloma steering committee, and hopefully we'll have a positive review and then be able to make this available to your frail patients to see which 3-drug regimen will be the best, since we expect to some extent that 4-drug regimens in this patient population will probably not be feasible from a safety perspective. And that actually is the last of our studies to mention, and wanted to therefore open up the session to anybody who had questions or wanted to make some comments about the previous presentations that you saw. If not, then I wanted to thank all of you for joining, and again wanted to thank all of the great statisticians and the vice chairs and our executive officer, and once again especially Jack Aiello on the patient advocate side. And we'll give you back about 25 minutes of your day. Enjoy the rest of the SWOG meeting and have a great fall. And we will not have a monthly call in October, so we do hope to join you on the call in November. Thanks very much and take care, everyone.
Thank you, everyone.
Thank you.